Before we start recognizing cells, first of all we have to look at the staining. How the smear stained. As you see here, we do have the red blood cells stained a little bit blue-gray, meaning that everything is more basophilic in this staining. The next one that you have to look at, or have to compare the size of the cells that you are interested in. For example, the middle, we do have a huge big cells. How can we measure the size of the cells? Let's compare to the red blood cells. We do know the size of the red blood cells around 7-8 micrometers. As you see here, that means these cells about at least 60 micrometers or 50 micrometers are the hugest cells. And after, you can see that this cell has multiple nucleoli inside. And in the cytoplasm, we do have some cloudy particles. Now these particles and the edge, you do see that is liberating some platelets. Now that's bigger cells, it's called megakaryocyte. The next staining is much better comparing to the previous one. Here the color of the red blood cells is brown or yellow-brown. Let's look at immediately these cells in the middle. These cells has a round nucleus. Comparing to the others, they do not have round nucleus. As you remember what we discussed at the beginning, the round nucleated cells mostly belongs to the erythroid cell line. Now, if you're sure that this cells is an erythroid, now we have to place the cells in the maturation line. Because this nucleus almost occupies the whole cell. It has a very narrow, teeny tiny cytoplasm, blue cytoplasm. The chromatin structure is homogeneous one. And we do have some darker stained nucleoli inside the nucleus. So this cells is an early erythroid precursor, such as a proerythroblast. If we place these cells into the erythroid cell line, you will see immediately this is very, very similar to the proerythroblast. Now, right next to this proerythroblast, we do have a different cell. The color of the cytoplasm is pale. We do have a very fine, cloudy granulation. The nucleus is oval-shaped and this is not round at all, and that the size about 16 micrometers, so about the same size as the granulocytes. So this cells is a myelocyte. Going to the left, there are two myeloid cells as well. These myeloid cells are bigger comparing to the previous one, and they do have some azurophilic granulation inside the cells. So these two cells we call as a promyelocytes. Other cells here, for example, the band neutrophilic granulocytes, and here we do see another cells with a basophilic cytoplasm with the almost round or elliptoid nucleus. That's the lymphocyte. Going up here, some orthochromatic normoblast and segmented neutrophils surrounds a cell. This cell has round nucleus located eccentrically, and the cytoplasm is basophilic, and the cytoplasm does have some empty holes, some vacuolation. So this cell is a plasma cell. Now, looking at these little eosinophilic spots, these are platelets, aggregated platelets. This is very, very important because based on the number of the platelets, you can conclude whether the number is less or more. However, is more accurate to measure by the CBC. This staining is better than the previous one. Looking at immediately, this cell, this is an erythroid cell with round nucleus. However, comparing to the chromatin structure to the previous one, this is not so homogeneous. You do see some precipitation already started here. But the cell is still big. It still has basophilic cytoplasm. But this is not the earliest one. this is the next stage, such as the basophilic erythroblast or basophilic normoblast. When you come down, this cell is still as erythroid cell, but this is smaller one compared to the basophilic normoblast, but not as small as the red blood cells. This is why it's a polychromatic normoblast, and this is an orthochromatic normoblast. In the middle here, we do see a lymphocyte, a small lymphocyte. Other cells belongs to the myeloid cell line, such as bean-shaped metamyelocytes or the band-shaped neutrophilic band and neutrophilic band here as well. This smear or this picture was taking an area where we did have the all erythroid maturation stages together. Here you can see the biggest one, such as the proerythroblast, the smaller one as a basophilic normoblast, and next one is a polychromatic normoblast, and the small one, this fourth, is a orthochromatic normoblast. Here you can follow how the maturation stages looks like and what are the difference between these stages. On this slide, we do have a different cell. We can see that the chromatin structure is homogeneous, however, it's not round and not as basophil as the previous one. So this cell is not an erythroid cell. This cell is a myeloid cell, and when you do see closely, this cell has lighter stained nucleoli. So that's a myeloblast, surrounded by segmented neutrophils and an orthochromatic normoblast right here. Very similarly here in the middle we do have a myeloblast surrounded by neutrophilic segmented orthochromatic normoblast, polychromatic normoblast, a lot of myeloid matured cells here, some lymphocytes as well, and here we can see a cell in division. The chromosome is going to be pulled in the two poles of the cells. Now this smear stained very dark blue, very dark gray. As you see here, mostly myeloid cells we do have. Maybe this cell is only one that has a round nucleus, that's an orthochromatic normoblast, but others are myeloid, except this, that's a lymphocyte. The earliest cell can be this, and that could be the myeloblast. And the next one in the top here, it still has a very blue cytoplasm. However, we do have several granules inside the cells. So that's an early promyelocyte. Next one, we do have the myelocyte, a lot of myelocyte. We do have band and segmented neutrophil granulocytes. This smear stained nicely. The earliest erythroid precursors can be seen here. That's a proerythroblast. All characteristic features can be seen. Very nice round nucleated, surrounded by lighter perinuclear area, basophilic cytoplasm, very homogeneous chromatin. The next one, as you see here, they were the same size or maybe a little bit smaller one, but the chromatin is getting a little bit more dense. So that's a proerythroblast, or that can be a late proerythroblast because the chromosome or the precipitation of the chromatin structure is not as much as it should be in the basophilic normoblast. On the top we do have a promyelocyte surrounded by neutrophilic segments, and here we do have a eosinophilic segment. We do have here an orthochromatic one, orthochromatic normoblast, possible that's a lymphocyte here, and some myelocyte, for example, and metamyelocyte. In this slide, in the middle, we do have a nice cells, round, eccentrically located nucleus, basophilic cytoplasm with vacuoles. For sure, that's a plasma cell. Other cells are neutrophilic segment, maturated segment, very few orthochromatic one. We do have a myelocyte stage, for example, eosinophilic myelocyte. Here possible that's a lobulated nucleated cells, that's a monocyte. Yes. Eosinophils. Eosinophils can be seen very nicely because they do have this brown, yellow, darker or bigger granulation. In the middle, we do have a very interesting cell. This cell is pretty big one because the size about 20 micrometers, and that's belonging to the myeloid cell line with a very dark and big granulation, and that's the basophilic myelocyte, surrounded by neutrophilic myelocyte, eosinophilic segment, but instead of having two, we do have three segments, so hypersegmented eosinophilic granulocytes. We have a neutrophilic band granulocytes, several segmented one, several lymphocytes surrounding these cells. This cell, similarly to the previous one, that's an erythroid precursor, an early precursor, that's a proerythroblast, very, very blue comparing to the other cells. Round, it's very light, narrow perinuclear area. That's a proerythroblast, with surrounded with lymphocytes, and this can be a band neutrophils or segmented neutrophils, or that's possible that's a myelocyte. This cell is not an erythroid cell because erythroid cells do not have granulation. This has blue cytoplasm but filled with granulation, a specific granulation. So this cell is called as a promyelocyte. Next to, we do have an orthochromatic normoblast and a neutrophilic segmented granulocytes. And here we do have a band or a late metamyelocyte. Very similarly, in the middle one, we do have a proerythroblast, eosinophilic myelocyte. This is a switched or twisted band, neutrophilic band, very similarly to this band, orthochromatic normoblast. Again, this picture was taken next to the previous one. That's a proerythroblast with this band or metamyelocyte. And that's a myeloid cell, but not the earliest one because we do have granulation in it. So that's called promyelocyte. This is an orthochromatic right here, and that's the segmented eosinophilic granulocyte. Looking at this cell, we do see some lobules in these cells, has very dark blue cytoplasm. So that's a monocyte cell line. This possible is a monoblast or monocyte. This is a lymphocyte, the same size as a red blood cell. Here we do see the polychromatic normoblast. Similarly to the previous erythroid cell and some other myeloid, possible myeloid cells, we do see polychromatic and orthochromatic normoblast. That can be a lymphocyte. Here mostly we do see the myeloid cell line, except this, that's a proerythroblast at the corner. Others such as this promyelocyte. Others are myelocytes, lymphocytes, orthochromatic one, metamyelocytes. And here, this cell possible, that's a monocytes or monoblast. Here we do see several cells that belongs to the myeloid cell mostly, and we do see an erythroid on the top. This erythroid, that's an early erythroid, that's a proerythroblast surrounded by this orthochromatic one, that's possible as a lymphocyte. Here we do see other orthochromatic one. Here we do have a polychromatic normoblast. In the middle, we do have an eccentric located cell such as a plasma cells, orthochromatic cells, Here we do see promyelocytes and myelocytes, band and segmented neutrophils and lymphocytes. Again, in this area, we do see this proerythroblast. That's a polychromatic one. Here we do see possible that's a myelocytes, these two. That's a lobulated one, can be a monocyte, and another are neutrophilic band and segmented one, several orthochromatic surrounded by. In this side, we do have two blue cells. One on the left, that's an erythroid precursor with light nucleus. That's a proerythroblast surrounded by polychromatic and orthochromatic normoblast. And we do have here an eosinophilic myelocyte. And the opposite, we do have a myeloid early precursor. There's no granulation with lighter nucleoli. That's a myeloblast. And several neutrophilic band, segmented one, lymphocyte. This is going to dominate the picture. On these slides in the middle, we do have a huge, big megakaryocytes. Inside the cytoplasm, we can see very nicely the different degraded platelets that by the edge, it's usually liberated out. These megakaryocytes are surrounded by erythroid precursors such as a polychromatophilic normoblast or orthochromatophilic normoblast. Not only the erythroid, but we can see the myeloid stage as well. That should be an early promyelocyte, so the late myeloblast, because if you're looking, you already see several granules inside the cytoplasm. And the myeloblast does not have granulation. So when the granulation is starting, we can call it as a promyelocyte. On the top, we do see an eosinophilic segment. You can see very nicely the two segments. A little bit down, we do see a lymphocyte, a small lymphocyte right here. Possibly this is a small lymphocyte as well. Several neutrophils segmented. Granulocytes, for example, myelocytes in the bottom, or promyelocytes surrounding by polychromatophilic normoblast. The next, we do see several components of the hematopoiesis. As we saw before, this is a basophilic myelocyte. Next to it, we do have a small cell. The size is very similar to the erythroid cell. So that's a small lymphocyte. Next one, this is an eccentrically located nucleus with a basophilic cytoplasm. That's very similar to these cells in the middle with vacuolation. So these two cells are plasma cells. Next to these plasma cells, we do have a round nucleated cells. So this cell as a proerythroblast is very early. You can see very nicely how homogeneous the chromatin structure. Similarly here on the top, that's a proerythroblast as well. All the way down here, we do see again an erythroid precursors. However, when you compare the chromatin structure to the previous one, this is a little bit precipitated, size smaller one. So that's a basophilic erythroblast or normoblast. Going to the right, we do see two polychromatophilic one and several orthochromatophilic normoblast. Here on the top, we do see a eosinophilic myelocyte. Here we do have two promyelocytes, and on the right we do have several neutrophilic granulocytes, segmented one band, and metamyelocytes. And here we do see a cell in division. The following slide contains several stages of the erythropoiesis. In the middle, we do have a proerythroblast. surrounded by polychromatic and orthochromatic normoblast. Here we do have a plasma cell and several myeloid cells, such as the band formation or myelocytes, or an early promyelocytes, late myeloblast, metamyelocytes, and few lymphocytes as well. On this slide, we do have in the middle two plasma cells side by side, surrounded by myeloid precursors such as a metamyelocyte, band, segmented one, and a few polychromatophilic one and orthochromatophilic normocyte. And here we do see at earliest erythroid precursors such as the
erythroblast.